All right, so we're still in the week of January 25th through the 27th, chapters 2 and 3. So we're going to finish 2, start chapter 3, finish chapter 3 next week. Next week you'll have exam 1 over chapters 1, 2, and 3. All right. All right, so lipids. Lipids are the most abundant, or I should say the most abundant lipids, rather, are your triglycerides, your phospholipids, and your steroids. Remember, we already covered carbohydrates. Now we're moving on to another class of organic compounds, the lipids. So, all right. Roll this toward me so you can hear on the recording. All right. So, lipids are going to be made up of carbon, hydrogen, and oxygen. And you'll have more carbon and hydrogen than you do oxygen. All right. Lipids are typically insoluble in water. Remember we talked about things being hydrophobic and hydrophilic? You know, hopefully y'all remember those two terms. Hydrophobic means they have a fear of water, don't like water. All right, and lipids are hydrophobic. Because remember, water is polar, and we'll see the lipids are mostly nonpolar. But they can be soluble in other lipids, so they can dissolve in other lipids. Okay. Where's my... All right, so some examples. You have the neutral fats or triglycerides, the phospholipids, the steroids, in which cholesterol is one. You have the bile salts, vitamin D, your sex hormones, and corticosteroids. Those are all steroids. Okay, not just the ones that people hear about taking to make themselves, have large muscles. These are all different steroids, and we'll see why. All right. So you also have your fat-soluble vitamins like A, E, and K. Also have the prostaglandins, the lipoproteins, and the glycolipids. All right. Common lipids in the human body include things like the triglycerides, the neutral fats. See them in fat deposits. Why they're good for your body, or is that they're ways your body stores energy. Okay, and they're built of fatty acids and a glycerol molecule. Okay, we'll learn about the structure of all of those. Okay, and you may have heard of these, but they are saturated fatty acids and unsaturated fatty acids. Anybody heard of saturated fat and saturated fat? All right. So that's what we're talking about here. All right, so here is an example of a triglyceride. And here, I'm not expecting you to memorize the structures, okay? I just want you to generally understand, what's going on here. So this is a glycerol molecule. You have 3 amino acids, or sorry, we're not there yet. All right, so I've got the noise out here that's distracting me. All right, you got 3 carbon atoms. So 3 carbon atoms in these OH groups. So that makes this glycerol. All right, anytime you see those 3 carbons like this, the OHs, it's glycerol. Then you got 3 fatty acid chains. So it's 1, 2, 3. Makes it, when you put them all together, you get a triglyceride. This happens through a process that I think we've talked about in here called dehydration synthesis. So here we're losing water, so it's dehydration, synthesis because we're making this thing, triglyceride. All right, and these are these long non-polar tails. All right. All right, let's see. All right. So looks like we just went through that. All right, let's talk about the difference between saturated fats and unsaturated fats. So you've got the saturated fats, and there you have only single covalent bonds. Okay, remember we learned about covalent bonds where the electrons are shared? Okay, hopefully you kind of vaguely remember that from last week. The chains are going to be straight because they're only single covalent bonds. And these can exist as solids in room temperature since the molecules pack closely together, which is generally not a good thing if it's inside your body. You don't want it to be solid, you want it to stay liquid. If it turns solid in your blood vessels, what's it gonna do? Clog up your arteries. Okay, another way to think about it is, when you say something is saturated, it's like it's wet, right? So saturated fats are saturated with hydrogen atoms. Okay, and that's why they have all single covalent bonds. Unsaturated fats have at least one double covalent bond. Okay, and it causes the chains to form a little kink, okay, or a little ridge. And these are going to exist as liquids and oils at room temperature. These are considered heart healthy. Why? Because they tend not to clog your arteries, all right? All right. So here in AMP, we're going to study something really, really fancy. It's called a stick of butter, all right? So there's your butter, and it's existing as a solid, a liquid, or a gas. What is that stick of butter? Is it a solid, liquid, or gas? Solid. Solid. Okay. All right. Now, so at room temperature, this is a solid because it has all single covalent bonds in those fatty acid chains. All right. And so they're all basically the same kind of basic shape here. So what do? They just stack on each other. It's kind of like if you have the chairs where you can stack the chairs on top of each other because they're the same type of chair. They just stack on each other. It's great for chairs, but it's bad for your body if you have fat, stacking on top of each other. So that's what saturated fat does. Now, I'll show you oil, right? So if you may use some oil for salad or things like that, right? So, so maybe some olive oil or things like that you may cook with it. All right. So these chains, these have all single covalent bonds. What's this right here? What's that one, the third one down here at the bottom? What's going on there? It's bending down. Why? Because it has a
Because it got unsaturated fat?
It has an unsaturated. Well, what makes it unsaturated? What does it have in it that makes it unsaturated?
Oil.
Well, it is an oil, but what on the structure? What's different? What's there on the third one is not on the other. The double covalent bond. Okay. It bends that structure. Okay, so it doesn't pack nicely anymore. That's what keeps it liquid. So that's what makes unsaturated fat unsaturated. Okay, those double covalent bonds. So your olive oil is olive oil. It's liquid because it has double covalent bonds. Okay, does that make sense? Whereas your butter is solid because it has single covalent bonds. All right. Now some more stuff that you may have seen in the grocery store. Anybody ever heard of trans fats? Anybody heard of omega-3 fatty acids? Okay. So the trans fats, these are oils that get solidified, become a solid, when you add hydrogen atoms at the double bond sites. And as you might expect, if you make things solid, is it good for your heart or bad for your heart? It's bad. It increases the risk of heart disease. That's why on those boxes of butter and other things it says no trans fat, right? Because they're trying to tell you it's more heart healthy. Okay. All right. So omega-3 fatty acids, where do you find them? Find them a lot in fish, cold water fish, as well as some plants. Some seeds like pumpkin seeds, chia seeds, flax seeds, walnuts, soy foods have them as well. These tend to be good fatty acids because they tend to decrease your risk of heart disease. Okay, so you tend to want the omega-3s, you tend not to want the trans fats. Okay, does that make sense? So you didn't know you were doing chemistry when you went to the grocery store, but you actually are. All right. Sorry to ruin the grocery store for you, but there's some chemistry there. All right, so common lipids in the human body. You also have phospholipids, all right? And these are not triglycerides. These have two fatty acids, okay? Because tri means three. These have two fatty acids. And they're going to have a head that has a phosphorus on it. So that's where the phospho and phospholipid comes in. And it has an electrical charge, okay? So it's polar. Remember we learned about ions, anions and cations, right? So it has a charge to it. It's polar. And because it's charged, it likes water, which is also polar, right? So the head's going to like water. The fatty acid tails or chains, remember those are nonpolar, so they don't like water. All right, and so phospholipids are critical to your bodies because they form your cell membranes, your plasma membranes, okay, which basically means they are the outer shell of all your cells, okay? All right, so this is a phospholipid, okay, so this to me looks like a high chair that's been turned on its side, fallen over, all right, had a broken leg or something. All right, but basically you have your polar head, has your phosphorus, all right, it has a charge here, you can see the N plus, there's a charge. Then you have the glycerol backbone, but you only have two fatty acid chains and they are nonpolar. And what do you know? One of them has a bend to it. Why does it have a bend to it? Because it has why is it one of those tails bent? We just talked about it. Double covalent bond. All right. But all this chemical stuff is the same as this little drawing over here. Okay, polar head, nonpolar tail. All right, that's all it is. So don't get bent out of shape about it. All right, common lipids in the human body. So lipids in the human body are going to include things like steroids. And steroids, like I said, are not just the ones that make people have big muscles. Okay, there are other steroids too. The thing that makes a steroid is that they have 4 interlocking rings. All right, if you see 4 rings together, it's a steroid. So cholesterol is a steroid, bowel salts are steroids, vitamin D is a steroid, some sex hormones are steroids. So how do you get these? Well, you get some cholesterol from your diet, you eat them, particularly if you're eating animal products, right? Your liver can also produce cholesterol. Why is cholesterol important? Because it's the basis for all your steroids that you make in your body. Okay? It's your starting material. All right. So what you see there in yellow? What's there in yellow? What do you see in the picture that's in yellow? What do you see there? Well, don't worry about the actual elements.
Somebody, I see the lines, maybe the double covalent.
You see a double covalent bond, that's correct. But what I also want you to see, why are the ones in yellow and yellow? The 4 what? The 4 interlocking rings. Okay, so these are the 4 interlocking rings that make a steroid. Okay, so if you see those 4 rings, it's a steroid. Okay, you don't have to know, oh, that's cholesterol, or that's vitamin D, or that's testosterone. You just have to know that that's a steroid. Okay, does that make sense? All right. All right, so before we move on to proteins, I want you to talk with your battle buddy about 3 things each of you know or has learned about lipids. And you can do this at home as well. So let's talk a little bit about proteins, next class of organic molecules. Okay, so we've already gone over carbohydrates, we've already gone over lipids. So proteins are pretty important. They make up over half of your body's organic matter, okay? And they're going to provide for basically the construction materials for your tissues, okay? And they are critical for how your cells work or how they function. Some roles that they play, proteins can be antibodies, help you with immunity. They can be hormones, they can send chemical signals around your body. And they can be enzymes, okay? And they can catalyze chemical reactions. All right, proteins are typically going to have, they are going to have these 4 compounds. They're going to have carbon, oxygen, hydrogen, and nitrogen. Occasionally you'll see some sulfur, okay? But at least for the carbon, oxygen, hydrogen, and nitrogen, you could remember it as CHON, or you can remember it as HONC, HONC. All right, but those are pretty important. And I guess if you're going to add sulfur, it becomes HONCs. All right, so and proteins are polymers. All right, so proteins are made up of monomers called amino acids. Okay, so just like your polysaccharides were made up of monosaccharides, Y'all remember that? Okay, your proteins are made up of amino acids. Okay. All right, so now that we've introduced the proteins, let's talk about their structure. So structure is that they are basically going to have a carbon atom in the middle, and then around it, they're going to have what we call an amine group, which is NH2. They're going to have an acid group, which is COOH. And they'll also have a hydrogen atom, and then they'll have what we call an R group, or a variable group. Okay? You have 20 different variable groups, or R groups, so you get 20 different amino acids. Okay, we're not going to have you memorize the structure of all the amino acids. We just want you to understand the general structure. Okay, so here's some examples. Here's your general structure here on the left. All right, so you have your generalized structure. You got your central carbon atom, you have your amine group on the left, and your acid group on the right. On the bottom, you have a hydrogen atom, and on the top, you've got the R group, the variable group. All right, so next to it, you have glycine. All right, everybody say hi, glycine. Okay, y'all didn't do it, but that's okay. All right, so glycine has a hydrogen as its R group. Okay, so then aspartic acid has the CH2 and then COOH as its R group. Lysine has a longer chain as its R group. And then cysteine has CH2 and an SH, sulfur and hydrogen. So that's where those sulfurs come in. Okay? So again, I'm not asking you to memorize all of those R groups. Okay? You just want to look at this general structure and be like, oh, there's an amine group, there's an acid group, and a variable group. Okay, that's an amino acid. That's what I want you to be able to do. Does that make sense? Okay.
So is the amine always gonna be on?
Yes. Generally speaking, I mean, 3 dimensions, it's kind of however you're looking at it, but for the sake of the chemical reactions, it's typically going to be expressed this way. Because of the way that the amino acids are going to link together, which what we're going to talk about next. Okay, protein structure. So polypeptide is a name for a protein that has fewer than 50 amino acids. So if it has 50 or less, polypeptide. And typically when I say 50 or less, it's really like 3 to 50, okay? Large proteins will have more than 50, but it could be as many as thousands of amino acids. All right, and just from one sequence of amino acids, you could actually produce a variety of different proteins depending on how it folds together. All right, now you need to know about the levels of structure. It's 4 levels. So primary. So primary is your sequence. Okay, primary structure is your sequence. Whatever the amino acids are, that's your primary structure. Your secondary structure has to do with kind of how it folds or moves on each other on itself based on the interaction of the atoms. And it can form an alpha helix, which is basically just a spiral, okay? But we call it an alpha helix. Or it can form, the protein can form a beta-pleated sheet. And a beta-pleated sheet, just to give you an example, is if I took this paper and let's just say I want to put some folds in it. So if the protein basically forms kind of lines back and forth, but they interact in such a way that it folds kind of like this, we call it a beta-pleated sheet, okay? Then you have tertiary structure. So if, you know, primary is first, tertiary or secondary is second, what do y'all think tertiary means? Third. So I'll help you to remember that tertiary structure is the three-dimensional structure. Okay, it's like, and so when we talk about the helices in the sheets, we're really talking about one section of the protein that forms those. Now on the three-dimensional structure, we're talking about the whole protein, all right? And quaternary structure is like the fourth. And the fourth dimension or the fourth level of structure means that you've got essentially multiple proteins that come together and it works as a complex, okay? All right, so I know it's kind of hard to visualize, so let's just kind of show you in pictures. All right, here's a chain of amino acids that have been bound together. That's your primary structure, okay? It's just your sequence of amino acids. Okay, we good there? All right, here's your secondary structure. There's your alpha helix on the left and your beta-pleated sheet on the right. Okay, can y'all see how the beta-pleated sheet, it looks kind of like a folded sheet of paper, the way that the atoms interact with each other? Now, what's causing the interaction? Do y'all see those little dashes? Y'all see those? Anybody know what those dashes represent? It tells you on the page. Hydrogen bonds, okay. See how things start building and we kind of link things we learn to new things? Okay, so that's what's gonna happen the whole course. You're gonna learn something and you're gonna apply it in a new context, okay? All right, but that's secondary structure, the alpha helix in the beta-pleated sheet. All right, here's tertiary structure. So in tertiary structure, again, this is not just the section like secondary structure. This is the whole thing. And see, in this protein, you actually have some sections that are beta-pleated sheets and other sections that are alpha helixes. Do y'all see that? And then other sections that just, you know, they move in other ways, essentially. They don't have that structure. But the whole three-dimensional structure is your tertiary structure. All right. And that leads us to quaternary structure. So quaternary structure, here you got multiple subunits. So the tertiary structure, you had one subunit. In this protein, you have 4 polypeptide subunits that come together to make it work. Okay. Hemoglobin is an example of that. Hemoglobin takes 4 subunits, puts them together so that your blood cells can carry oxygen, okay? Okay, a little more on proteins. You have fibrous proteins, also known as structural proteins. They can appear in different body structures, and they can exhibit tertiary, secondary, tertiary, or quaternary structure, okay? So they can have the sheets of the folds, they can be three-dimensional structure of the whole protein, they can be multiple subunits. All right, and their role is to connect structures together, and you'll see them in your body's tissues. They're very stable. Examples of structural proteins are proteins like collagen and keratin, and we'll learn more about those when we study the integumentary system. Okay. All right, so there's your collagen. It's a triple helix. Okay, so here, that's going to be very strong. It's three strands together. All right. So it's structural, it's fibrous. All right, globular. Let's see. There we are. You have the globular or functional proteins. So these exist as enzymes. They have a function, catalyzing reactions, hormones, chemical messengers, and antibodies that help you with infections, or fight infections. They're going to typically exhibit at least some level of tertiary structure, which means they could exist as one subunit. Okay? Could be multiple subunits. And they can be what we call denatured. And basically means, y'all saw that three-dimensional structure in the tertiary structure. If you heat it up too much or you get some chemicals on it, it destroys those hydrogen bonds. And then it doesn't fold the way it's supposed to fold. Okay, so it gets denatured. And so it can't perform its physiological role, can't perform its body's function. All right, now, if it's an enzyme, the enzyme is going to have what we call an active site. All right, so the enzyme, I like to say it this way, the chemical reaction is the party, the enzyme is the party planner. Okay, it gets all the parties together for the conditions to happen, right? So, so if you're the negotiator in a business transaction, you have to get the parties together in order to come together to form some agreement. And that happens at the active site on the enzyme, all right? All right. So this is hemoglobin protein. So you have the globin protein subunits. So these have a function, they're globular. And then you get the heme group that's in there mixed in, that makes your hemoglobin molecule. Okay. But when they have a function other than just providing support, they're typically globular. Okay. All right. Functional proteins, again, antibodies or immunoglobulins, help you with infection, the hormones, regulate growth and development. Transport proteins can transport oxygen like hemoglobin does or other materials in your body. All right, enzymes, which catalyze chemical reactions, make it so that it takes less energy for those reactions to happen. Okay, speaking of enzymes, so they are biological catalysts, which means that they're not used up. Okay, so you can use them and use them and use them. Okay, they don't get broken down. They're not products. All right, they're not reactants and they're not products. They don't get changed. They increase the rate of the chemical reaction. It happens more quickly. Okay, and they are binding to what we call substrates at the active site. Remember I said that the active site's where the party happens. Okay, so they bind those starting materials, those reactants, to the active site of the enzyme. And that's where that reaction happens. And if you see the suffix, remember suffix comes at the end of a word in English class? You see ASE, it's an enzyme. Example here, hydrolase and oxidase. These are enzymes. So you'll learn a lot of root words in the class. So that's ACE is one example. All right. So the whole thing about the enzyme, I think sometimes it's hard to understand. I think pictures help out a lot. So here's your enzyme. And it has, what we said, it has an active site. They're on the left. Okay, the substrates are the amino acids in this case. All right, so those amino acids fit onto the enzyme. And they form what we call an enzyme substrate complex because there's enzymes, there's an enzyme there and there's substrates there, right? Complex means you have things together. Then you've got some reaction occurring. You're absorbing energy and forming a bond and releasing water. Okay, once that peptide bond's been created, you now have a protein. Protein's ready to leave the enzyme. The enzyme's not used up. Another set of proteins can get in there and use that space. Okay, make more protein. So does everybody have a better idea what the active site is and what it does? It makes the reaction happen. Okay. All right, so let's take a break and talk to your battle buddy about characteristics of proteins and particularly about enzymes. Okay, so I'd like you to talk to your partner about the structure. So let's do it this way. One partner is going to talk about the structure and the levels of protein structure. And then another is going to talk about enzymes. Okay, we'll do that one. All right. You can do this at home as well. So now that we've discussed carbohydrates, lipids, and proteins, we can discuss nucleic acids. So what are they? So nucleic acids, what they do is they are what makes up your genes, okay, your genetic material, your inherited characteristics. They're made up of elements carbon, oxygen, hydrogen, nitrogen, and phosphorus atoms. They're the largest biological molecules that you have in your body, actually. Okay, they get very long. All right, so they are, there we go. So they're built from nucleotides. Nucleotides are the building blocks, the monomers for nucleic acids. And those nucleotides have three main parts. They've got a nitrogenous base, which means it has nitrogen in it, which was an element. And there are essentially 5 different nitrogenous bases. You have adenine, which is abbreviated A, guanine, which is abbreviated G, cytosine, which is abbreviated C, thymine, which is abbreviated T, and uracil, which is abbreviated U. Okay? Nucleotides are also going to have a pentose sugar. And just like pentagon has 5 sides, pentose sugar has 5 carbons. Okay, it also is going to form a 5-membered ring with one of those carbons sitting on the outside. All right, you also get a phosphate group, which is basically phosphorus and oxygen. Okay. Those are the components of a nucleotide, which are the building blocks of nucleic acids. All right, so deoxyribose. So deoxyribose is a sugar that we find in DNA. And it has, there's the phosphate group, there's the pentose sugar. Okay, we only wrote out the CH2 that's on the upper left-hand corner, but the other corners are also carbon atoms, okay? So, and then you got your nitrogenous base, adenine. So 1, 2, 3, 4, 5. Okay. All right. So now that you've seen deoxyribose, let's talk about DNA, deoxyribonucleic acid. So it's your genetic material that you have in the nucleus of your cells. And that's where you get your instructions for all your proteins in your body. Tells your body how to make all the proteins. And it's organized by what we call complementary nitrogenous bases. And it forms a double-stranded helix. So the bases on one strand are going to be complementary to the bases on the other strand. Okay? So it's kind of like sides of a ladder, where the two sides, the rungs are where the two sides are interacting with each other. Except this ladder spins around and forms a helix. Okay? And or think about it like, anybody ever seen a spiral staircase? Okay, it's kind of like that. All right. So it contains sugar deoxyribose, and the bases are adenine, thymine, cytosine, and guanine. Okay, remember we said there was 5 nitrogenous bases? Well, these are the 4 that are in DNA. And your DNA has to be replicated or copied before you go into cell division or mitosis. Okay? So here's an adenine nucleotide. Okay? Here's a computer-generated image of what DNA looks like. Okay, you can see it kind of rotates. Okay, it's one of the spiral, double helix. All right, this is sort of a picture version of it. Remember I said the spiral staircase? Okay, so each strand has nucleotides along it. Like here, A, C, A, you can see those, and then it spins around here, A, G, C. Now, these are the complementary nucleotides. All right, so T is complementary to A, G is complementary to C. Okay. And then along your strand, purple areas are light purple, lavender, that's your deoxyribose sugar, and your dark purple, that's your phosphate. Okay, so that's your backbone, okay? Now, we show you that to tell you that there's also ribonucleic acid, or RNA, okay? And this has come up a lot in the news because, you know, a lot of the coronavirus vaccines that are out there are what kind of vaccines? They are RNA vaccines, okay? So RNA is important as well. So it carries out the instructions from the DNA for protein synthesis. Okay, so DNA is sort of the master controller, tells RNA what to do to make protein. And RNA is made from a DNA template. Okay, so you use DNA to make RNA. It doesn't become RNA. You use DNA to make RNA. Okay, and it's organized by what we call those complementary bases, but it forms a single-stranded helix. How many strands do we say were in DNA? Two. RNA is just one. Okay. Contains the sugar ribose. Okay, so that's different because what was the sugar in DNA? Deoxyribose. Okay, so RNA has ribose. And then the bases, adenine, cytosine, guanine, and uracil. Okay, so that's a difference. So adenine, cytosine, and guanine are in both DNA and RNA, but RNA has uracil, DNA has thymine. Okay, it's an important distinction. All right, DNA has the bases ACG and T. RNA has the bases ACG and U. Okay, and there's three varieties of RNA. You have messenger RNA, transfer RNA, and ribosomal RNA. And we'll cover those more in the next chapter. Okay, ATP, adenosine triphosphate. So it's an energy source. Okay, it's your body's main source for energy. So why are we covering it here? Because it's made up of a nucleotide, right? In the nucleic acid section. So it's built from a ribose sugar, an adenine base, and 3 phosphate groups. Okay? It's a form of chemical energy used in your cells. And you release that energy when you break the high energy phosphate bonds. All right? So we'll show you what that looks like, but the energy is trapped in those phosphate bonds. And if you break one, you're releasing some energy. Okay, ATP, it's going to get replenished, so you can make more of it when you oxidize or break down your food. Okay? So that's why you have to keep eating, so your body can continue to make ATP. Or one of the reasons, anyway. All right. So here is a picture of ATP. So you have your adenine, nucleotide, ribose sugar, or the 5-carbon ring, or 5-carbon sugar, okay, and 3 phosphates. You see the 3 P's there on the left, on the upper left-hand corner? Do y'all see the 3 P's? Okay, those are your phosphate. And so there are high energy bonds between the phosphate groups, the little red squiggly lines. Now, if you take ATP and you hydrolyze it, and you use water to break a bond, you break one of those high energy phosphate bonds, you release some energy, and you're left with adenosine diphosphate, because now it just has 2 phosphates and a free-floating inorganic phosphate. Okay? It's inorganic because it doesn't have carbon and hydrogen, right? It's just phosphorus and oxygen. So as you consume your ATP and you use it to get energy out, you start accumulating adenosine diphosphate. Okay? We saw that in the chemical equation. All right. So we're going to show you some examples of how ATP drives the work in our cells. Okay. So you have chemical work, transport work, and mechanical work. So chemical work here, ATP is giving you energy to drive these chemical reactions that absorb energy. Okay, so you're forming a bond between A and B, so you're having to absorb energy to create that bond. And that energy came from ATP. Also, ATP is involved in transport work. So it can transport solutes across the membrane in the cell. Mechanical work. So here, ATP is activating these proteins that contract in your muscle cells. And they shorten, so that's mechanical work. Okay, so those are three types of work that ATP can help facilitate. All right, so that is chapter 2. So really you got 5 minutes, so we're gonna go ahead and do your exit tickets and we'll start chapter 3 on Monday. We're gonna have to go fast and furious so you can get, take your exam. But this gives you the foundation for chapter 3. All right.
Are exams on Monday?